thank you all for having me. It's always great to speak to Michigan folks. Full disclosure, though, I haven't fully recovered from the Michigan State football game, and I'm hoping nobody asks me any questions about that at the end. So Nigel asked me to talk about the CHEER program. So CHEER stands for the Child Health Exposure Analysis Resource Program, and it's a laboratory consortium, which is very unique. There aren't a lot of them in exposure science. In fact, I think it's the only one that I'm aware of. There are occasional laboratory consortiums for other fields like metabolomics. CIDR is a type of laboratory consortium in genomics, but this is really the only one I'm aware of in environmental health. So I think it's a very novel consortium, and it's also a very important one because it's starting to bring environment into a lot of different studies. So the importance of biomarkers is that they inform exposure and mechanism. Mechanism can inform prevention, diagnosis, and treatment measures. In CHEER, which was recently renamed as HERE, so instead of Child Health, it's now Human Health Exposure Analysis Resource, provides a set of biomarkers that informs the role of environmental contributors to health, development, and disease. So this is the current configuration of the HERE network. There are 4 components, actually there's 5 components. The first component is the targeted CHEER lab hub. So there are 3 targeted CHEER lab hubs, and I'll define what targeted means in a moment. I'm the PI of the Mount Sinai targeted lab hub. The Wadsworth Center is actually, the PI is Patrick Parsons. And at the University of Minnesota, it's Lisa Peterson. And we provide an array of panels that are for environmental exposures and also nutritional biomarkers. There's an untargeted CHEER lab hub. There's actually 2 of them. One is at RTI in North Carolina, and that's directed by Tim Fennell. And the other is also here at Mount Sinai, and that's directed by Manish Arora. And these are untargeted chemical assays, which I'll talk about a little bit later in the talk. There is a data center, which is also based at Mount Sinai, and Susan Teitelbaum is the PI of the CHEER, or the HERE Data Center. And the coordinating center is actually based at Westat, where Barbara O'Brien is the PI. And the 5th component of HERE, and this was a component that was added when we went from CHEER to HERE, is actually the Environmental Sample Lab Hub, which is based at Duke University, and the PI there is Heather Stapleton. So these are, for the Duke Lab Hub, these would be things like dust or air filters from air monitors that you want to analyze for inorganics, organics, or even non-targeted chemical assays. So how can environmental epidemiology address mechanism through CHEER biomarkers? Well, in the original program, there was actually, we used to do biological markers of response, so you could actually get epigenetic marks, mitochondriomic marks, even some genetics. But in the reconfiguration, when we went from CHEER to HERE, actually that part of the program was taken out. And now we're really just focusing on environmental exposure biomarkers. So, but that doesn't mean you can't understand or get some information about mechanism. I would argue that exposure timing gives you some information about mechanism. It's sort of an extreme example, the way lead works on the brain in a 2-year-old who's been exposed to lead is very different than the way lead works on the brain in a 75-year-old who's been exposed to lead. In one instance, it's actually affecting the way synapses and neuronal networks are being formed in the 2-year-old. And in the other, it may be accelerating neurodegenerative processes that have other initiators, but in which it's actually accelerating it. So it's a very different mechanism for effects that may even seem similar on the surface, but there is information in exposure timing. And then there's mixtures. So people think of gene environment interaction as a type of mixture, or excuse me, as a type of biomarker that explains mechanism. If the particular gene is shown to interact with an environmental exposure, that gives you some information on how that environmental exposure may work. Basically what you're saying is that environmental exposure occurs on a genetic background. You can also think of mixtures that way. So if you're getting exposed to lead, it's happening on an environmental background that's variable. So there may be host susceptibility factors in mixtures that we need to understand, and that may be why some people get exposed to lead and seem relatively resistant, and other people seem very, very sensitive. Some of that may be genetic susceptibility. Some of that may even be environmental susceptibility. I think mixtures gives us the ability to understand some of that variability that we see with risk factors for disease. And so CHEER or HERE can actually help with these two as well. So in terms of how CHEER is going to inform child health research, really the ultimate goal is to study the exposome. There's lots of definitions of the exposome out there. I think I try to avoid the word totality because I think it's not realistic. So I prefer to define it as all the health relevant human exposures from conception to death. And I think we may never get to all of them, but we can get closer and closer. And in fact, there has been enormous advances in the last 10 years in our ability to measure the exposome that I think a lot of researchers outside the field aren't aware of. And it's actually quite advanced at this point. And just as genomics started with small sets of genetic variants and grew to studying GWAS and whole genome sequencing, exposomics is starting with mixtures, and I think it's actually going to grow. In fact, there's a lot of evidence that it's going to grow. And in fact, the targeted assays that I'm going to talk about, you can think of them as the equivalent of microarrays in genetics, and the untargeted are the equivalent of more advanced, either higher order microarrays or even whole genome sequencing. In fact, oftentimes people who do untargeted assays talk about it in terms of sequencing the exposome. And I think exposomics right now may even be a little farther than 2003, but it's about where genomics was in 2003. I'm old enough to remember candidate gene studies when we had microarrays that may have measured 256 or 512, and then 1,024 SNPs, and that was a big deal back in 2003. And now if you're not doing whole genome sequencing or you're not doing a higher order GWAS, you're not really doing state-of-the-art research. But just 15 years ago, a few hundred SNPs was actually considered state-of-the-art. And I think exposomics is actually getting there. So now I think going forward, you're going to see studies where there are higher order mixtures and studies where perhaps even untargeted chemical assays are becoming more and more the norm. So this is how CHEER or HERE works. So the investigator must be NIH funded, and I put the investigator in the middle. In our conference calls, we often refer to the investigators as clients because we are a service lab, although we provide expert consultation on how to link your environmental factors to your disease. So if you have a study that's on a particular health outcome, we may steer you towards a particular set of environmental exposures. If it's neurodevelopment, it might be things like methylmercury or phthalates or lead, whereas if you're studying asthma, it might be phthalates or PAH or something that's more likely to affect the lungs. So we actually may steer you in a particular direction depending on the phenotype that you're studying. Also based on the biological samples that you've collected. You have to keep in mind that in order to apply to the HERE network, the biosamples must be in hand. So that means you can't apply for samples that you plan to collect. You can only apply for samples that you've already collected and are storing, which gives sort of a different dynamic to the consultation, because if you collected them in the wrong tube, that may actually change everything. So there are lots of little nuances to doing these consults. So I run a targeted lab hub, and the types of assays that we run are on the order of metals, pesticides, plasticizers, phenols, halogenated compounds, and that would include PFAS, vitamins and hormones. We also do some nutritional assays as well. The untargeted assays are more of a screen where they actually generate a chromatogram, which represents all the chemicals inside the biological sample. And there's probably a few thousand which we have the potential to identify, although in any individual person, they're not going to have everything in our library. So it's more on the order of 500 to 1,000 are clearly identifiable and quantifiable. But there are statistical techniques to actually analyze the chromatogram without actually knowing with great certainty what the particular chromatographic feature represents. And that's a way to sort of do some data reduction to figure out which of the chromatographic features are important. And then you can try to zero in and figure out what that chromatographic feature actually is in terms of what that chemical is, if it turns out to be the biggest hit associated with a disease. There's a data center that will help you do analysis on mixtures, but also analyzing untargeted assays as well. And then the expert consultation is really an iterative process between the data center and the lab hubs. And both the targeted and untargeted lab hubs have development cores. So we're always developing new assays that we're bringing online. So if you look at our website today and look at it in 2 years, you might see some additional types of assays being added to the website as they come on board. So this is an overview of the CHEER Lab Hub resources. And as I mentioned, we used to do biological response assays, which were things like gene expression and DNA methylation. In our current iteration, we don't do that anymore. So we really only do targeted analysis and untargeted analysis, and we develop new assays. So in terms of the targeted analysis, we can measure organics and elements in whole blood, plasma, urine, hair. I'm going to talk to you a little bit about teeth towards the end. Actually, many of you probably know Dr. Manisha Arora. Many of you may even collaborate with Dr. Arora, who's developed a method to actually analyze teeth and go back in time to reconstruct past exposures. Similar methods can be used for other soft tissues, such as tumors or placenta, to actually get a geographical or a geospatial estimate of where the chemical is or the element is in a tissue. So it may not be that the total concentration predicts a health outcome. It may be the way it's distributed in a tissue, a particular chemical that predicts a health outcome. So that's a different way of thinking of exposure in more of a two-dimensional or even three-dimensional space. And then the untargeted assays are not just chemicals. You can also get the lipidome. We can also do proteomics. We can do adaptomics. We no longer do the responsomics, so in our new iteration without the biological response. NIH had some guidelines on what they would consider exposure and what they would consider response. Inflammation was sort of a gray zone, so we still do some inflammatory biomarkers, but we no longer do the immunoarrays. We also cannot measure DNA conformation, but in our development cores, we are measuring different chemicals in different types of tissues. Dr. Arora is working on organics in dental layers, so to reconstruct past exposure to past organic chemicals like phthalates and phenols. I mentioned the tissue mapping. We're also working on organics with that. And also looking at some unique matrices. We've done some work with umbilical cords, umbilical arteries, and umbilical veins as well. So CHEER and ECHO. So ECHO and CHEER are very closely related. In fact, the initial funding for the CHEER program was also from the National Children's Study funding. The requirement by law was that the work had to be dedicated to children's environmental health, which is why the initial lab consortium was called CHEER rather than HERE. We are no longer funded by that source, so that's part of why we changed our name, and our funding sources are actually different now. But CHEER measures exposure via assays. There was another program called PRISMS. To be honest, I don't know very much about it, other than it was also funded by the National Children's Study funds. And it was a program to develop wearable personal monitors for children. There are a number of sites that were funded. I've not heard much about it, and I've always been curious about what they developed over the last 5 years, but it's not something, to be honest, I know something about, but if somebody knows, I'd be happy to hear about it because I'm interested in it myself. So ECHO is a consortium of prospective cohorts. The ECHO RFAs included CHEER supplements that could fund assays, but that means there's a budget for assays. So it is not a sort of top-down approach to assays in that everyone's going to get a blood lead. There are applications, and obviously I think mostly people on the call know this, there are applications for analysis plans that request CHEER services that go through a process by which they may get funding approved to allow for those assays to occur. So as I said, our program CHEER turned into HERE in 2019, and our funding changed. So now we're funded through the NIHS budget, but also NCI and NHLBI contribute to HERE. The ECHO program still contributes to HERE. There is talk of other institutes joining in the future. In fact, NIDDK actually put out an RFA that was specific to chronic kidney disease of unknown origin. And in the RFA, they talk about using HERE for exposure assays. So we expect NIDDK to join the HERE program pretty soon as well. This is important because in order to apply for HERE, your study has to be funded by one of these programs, either NIEHS, NCI, NHLBI, or ECHO. Otherwise, you cannot apply for HERE funding. NIDA has actually expressed some interest because of the ABCD study, so that's another institute that may come on in the future, but right now it has to be for one of these four programs. And the ECHO funding is actually dependent on ECHO's allocation to HERE, which means there's a budget, which means there's a lot of decisions that have to be made before it gets approved. And unfortunately, some of those decisions are more budgetary than scientific. So what about the toxicological consultation? So I actually believe this to be one of the greatest services that we provide. A lot of people are coming into HERE who've never done environmental health. Their background might be in nutrition, it might be in psychology, it might be in genetics, and environmental health is a bit of a foreign field to them. So one of the things that we can do is help you understand what is possible. So remember, in every case, this is an existing study that has already collected its samples. So we're not providing advice on what to collect going forward. We're providing advice on what is possible, what is scientifically valid in the biospecimen archive that you already have. So the kinds of questions we can answer are, I have urine for mercury, it could be organic or inorganic. To answer the question, it's actually inorganic. Urine and mercury and urine is uniformly inorganic. And it does matter in that inorganic mercury is toxic to the kidney and methyl mercury is not. So it's a completely different form. So in terms of other things, if you want to study manganese, what would be the optimal biomatrix to use? If you have whole blood, urine, serum, or plasma, I would probably advise you to use whole blood, although serum and plasma would probably be reasonable. Second choice is urine. Manganese is actually excreted in the bile, so most of it actually does not show up in urine, so I would actually probably advise against urine in that case. And should all ECHO cohorts get measures of urinary DAPs? DAPs represent organophosphate exposure. Well, if you're living in, to bring it back to Michigan, if you live in Livonia or if you live in Lansing, it's probably not going to be very high, but if you live about 30 or 40 miles outside of Lansing in far country, it might actually be a reasonable thing to look at. So usually it's agricultural populations where we see organophosphates. So maybe you're interested in BPA, but you don't have urine, but you have serum. Is that okay? The answer is no. Serum BPA is very, very low, typically below detection limit. It gets concentrated in the urine. That's why it's a preferred biomarker. So it wouldn't be a particularly useful thing to look for BPA in serum. And that's an important question from the investigative perspective as well, because you don't want to waste your precious serum on an assay in which 90% of the samples are going to be below the detection limit. So that's actually, even though it's not an answer everybody wants to hear, it's a very valuable answer because it allows you to preserve your precious samples for something else. If you store urine at -20, can you use it for phthalates? Answer to that question is yes, although the number of freeze thaws can be important. If there's been no freeze thaws, it's not a problem. If there's been 4 or 5, we might want to do a pilot study to make sure that it's actually measurable. And if you have measured multiple metals in hair as a mixture and you want to combine them and use z-scores with measures in another study that use blood, is that really comparable? I would argue that it's probably representing a different point in time. Hair grows at about 1 centimeter a month. So if you've been using hair, you're actually looking at a cumulative exposure for several months back, which is a very different timeline than if you're measuring all of them in blood. And then tubes sometimes matter. If you collected plasma in a blue top tube, you've collected it using citrate as opposed to EDTA, which is in the lavender top tube, and that can affect the assay. And if you measure, or if you collected serum with an SST tube, these are sometimes called tiger top tubes, so the ones with the sort of gray and red caps. Those actually have a gel in them, and they have a hydrophobic coating on the inner surface of the tube that allows the gel to move. The SST stands for serum separation tube. And so the gel actually separates the clot from the serum. And it moves in order to do that. And that's why there's this hydrophobic coating on the inner of the tube. The problem is that if you're looking at organic chemicals that are very hydrophobic, they may actually bind to both the gel and the hydrophobic coating, and it may actually affect your results. So we try to avoid that. It may not be that big of an effect, but if you can avoid having that uncertainty added to your study, particularly if you have a plain red top tube, we would advise you to use the plain red top tube and not the SST tube. So those are just some of the examples of things that have come up in multiple HERE consultations in the past. All those examples are things that we've discussed with investigators. So our targeted assays. So targeted assays are kind of like microarrays. So you can think of that for the geneticists on the call. Microarrays can be customized, panels a little bit customized, but we're looking at dozens of chemicals in a panel that we can measure all at once. And they often have very similar chemical properties. We're not looking at one chemical, we're looking at dozens of them. And OnTarget is more like a GWAS or a whole genome sequencing. And then we can also do the same thing with nutrients. Nutrients can be minerals, so we can measure minerals, and we actually do have an elemental panel of about 20 elements that we measure, which is a mix of nutrients and potential toxicants. But panels can include fatty acids, they can include vitamins. There are also metabolic chemicals, such as hormones that we'll measure, and inflammation markers, such as DNA adducts or isoprostanes. And as I mentioned earlier, we can measure them in a whole host of different tissues: blood, plasma, serum, urine, hair, toenails, saliva, and teeth are all things that we've worked with, and there's other tissues that we're willing to work with as well. I anticipate, because NCI is now part of HERE, that we'll be actually measuring some chemicals in tumor tissue as well. So this is just an example of what we do at Mount Sinai. So our chemist, I want to give credit where credit is due, is Dr. Siam Andra. I say that and then I mispronounce his name. Dr. Siam Andra is our chemist, and he has developed a series of panels on organic chemicals, which are really advanced and are very, very interesting. So within these boxes, these are all panels, but he has methods to actually measure across different classes. And so what he'll do is, on our consultation, we'll get an idea of whether or not it's likely that we're going to find some of these chemicals. So neonicotinoids, as a good example, that's a type of newer pesticide. If your study takes place in an agricultural population, we may try to see whether or not we can find neonicotinoids. If it's very, very tropical and you're using a lot of mosquito repellent, we may try to look for DEET. But if you're living in Canada, we're probably not going to do that because it's unlikely that we're going to find it. But based on some pilot work that we'll do on your initial samples, we'll break down which panels are likely to yield good results, and we'll focus on those panels in the full CHEER or HERE assay job. And we can do the same with smoke. So these are the panels that he can do collectively if exposure levels are high enough. So that's why they're in these big red boxes. And then phthalates, you can do phthalates, phthalate replacements, and alternatives. In terms of serum or plasma, he has a multi-class persistent organic pollutant panel, which includes all of these chemicals. Being Michigan, PBBs may be of interest. There's also a panel for PFOS, PFOA, and all the different types of PFOS and PFOA. In fact, it's not represented on this slide, but actually Siam has developed an assay, which is an untargeted PFAS assay. So he could actually measure approximately 200 PFASs. So most of the PFAS assay panels that they're working with, it's about 10 to 15 chemicals. There's actually hundreds of different PFASs out there. Those very similar in structure to fatty acids. And so he's developing methods to actually expand the number of PFASs that can be measured. And then we also do nutritional panels, such as phytoestrogens, vitamins, folate, and mycotoxin, which is has a steroid backbone to it. And then we are doing some bioresponse assays, such as DNA oxidation, lipid peroxidation, and protein oxidation, which NIH is allowing us to do because inflammation and exposure are so tightly tied. They decided that that particular bioresponse assay was still part of HERE. And this is a very similar slide, and I've shared these slides with Tracy. So please, if you need them, please just get them from Tracy. We're welcome to it, just to give you a sense of what we can do. But all of those different chemicals that I talked about on the slides are also represented here by the boxes. The boxes are represented by the panel and class, and they tend to go together. So box one is all these can, in theory, be brought together, although we want to do some pilot work to make sure that they're actually measurable before we do it in the long run, because it takes up a lot more instrument time if we did all 15 of these panels at once. And if most of them are going to be below detection limit, we can reduce the amount of time we spend in the laboratory if we skip over the ones that are mostly below detection limit and just focus on the ones that are above detection limit. And then this is our elemental panel. There's roughly 20 elements on this. Some of them are nutrients, such as copper, manganese actually can both be a nutrient and intoxicant. And many of them are just toxic, such as mercury and lead. Some of them, like barium and strontium, are actually found in teeth. And they're also found in both breast milk and in formula. And they're found in very different concentrations in breast milk and formula. And one of our faculty, Dr. Christine Austin, actually has figured out methods to look at barium and strontium and reconstruct past breastfeeding patterns based on the amount of barium and strontium in your teeth layers. So we're also starting to look at breast milk and breastfeeding using the tooth biomarker and reconstructing it going backwards. We can measure fluoride, but that's a very different assay. It can't be done on ICP-MS, so it's not part of the panel. And iron is also not done by ICP-MS, so when it is done, it's done separately. So a typical workflow, whether you're doing untargeted or targeted, starts with whether you're first with your approach, separation, which can be typically either LC or GC. For untargeted, we typically now use an Orbitrap, although we do have QTOF capacity. Orbitrap is becoming sort of the industry standard in exposomics, at least, because it is much more higher resolution. So we're able to get more of the lower concentration chemicals. That's important because the exogenous chemicals like PFAS and PFOA and PBDEs can be detected with an untargeted assay, but only with the Orbitrap, because only the Orbitrap has a sensitivity to get chemicals with that low of a concentration. In untargeted, we're looking at features that we're extracting. In targeted, we know what we're looking for, so we have standards and we look for the particular formula based on the mass spec and the separation technique. And then we do some data reduction and sample processing before it gets submitted to the CHEER here data center. And as I said, now our gold standard method is almost uniformly using the Orbitrap high resolution mass spectrometer, although we do still have QTOF capacity. QTOF is very good for metabolomics, and it's also much faster and easier. But to get to those lower concentration chemicals, we find that the Orbitrap actually is superior. So this is an example of a chromatogram. So as I mentioned, the metabolites are probably the higher peaks, and the exogenous chemicals are probably the lower peaks. And you get more resolution on an Orbitrap for those lower and smaller peaks. So I'm going to spend the last part of the talk going over a couple of use cases to just give you a sense of how the process works once you've been once you put in an application, you start to interact with a lab hub. So let's say you have a longitudinal birth cohort study. So this could be, say, an echo cohort. You're looking at infant cognition and breastfeeding patterns. Women were enrolled at delivery. They collected diaries of breastfeeding habits going prospectively, and breast milk was collected every 2 months. There was some maternal plasma that was collected at the time of delivery. They were separated as plasma and red blood cells. The red blood cells were saved. Urine and hair were also collected at delivery. There were no biological samples collected going forward, however, but DNA was collected from maternal saliva at delivery, and there is cord blood in the children. And then the study looked at Bailey scales and, say, a facial recognition test at 1 and 2 years of age. Plasma was stored at -80, urine at -20, red blood cells at -20, and the hair was kept at room temperature. So in the consultation, if you were to come to us in CHEER and here, we would ask what samples were bioarchived, how long were they stored, at what temperature, and for how long? Did you aliquot them? And if so, how big are the aliquots? It's always better to aliquot because you go through fewer freeze thaws if you have aliquots. We usually recommend about a 1 to 2 milliliter aliquot. 2 is better for urine, 1 is probably better for plasma, and that'll reduce the number of freeze thaws you have to do if you don't have to go back to the same big sample all at once. So what research question is of interest matters. So in this study, you're looking at infant cognition. So we would tend to focus, for the targeted assays at least, on chemicals that we think are biologically plausibly related to infant development. And then thinking about the ADME, ADME stands for absorption, distribution, metabolism, and excretion. It's a pharmacologic toxicokinetic term, but it does matter because the toxicokinetics of hair versus plasma and versus urine are quite different, and for the same chemical may actually give you very, very different information. So you want to be able to use that information in your analysis and help you choose which biological matrix you want to use. So some examples from this example, or some advice that we might give for this example. Well, metals actually bind to hemoglobin really effectively. So 99% of lead in the blood is actually bound to hemoglobin. So red blood cells tend to be really rich in metals. The same is true for mercury. So if you have those red blood cells, there are some things you can do with them. You can measure hemoglobin A1C, but you can also measure metals. And in fact, because it's 99.9% of lead and mercury are bound to the red blood cell, if you actually measure the hematocrit, you can actually reconstruct what the blood lead and the blood mercury level was with that information. Mercury is actually secreted into the hair and it's also excreted into the bile. It does not go out in the urine. But if you have hair, methylmercury is actually quite a valid biomarker in hair. People always worry about contamination with hair, although I think hair probably has a lot more value than a lot of researchers realize. Certainly if it's a metabolic product, contamination should not be an issue. So you don't get contamination with cotinine. Cotinine is a breakdown product of nicotine. So any cotinine in the hair isn't from external contamination. The same is true if you measured hair cortisol. Also, there are ways to clean hair pretty rigorously so that you're actually only looking at the interior concentrations of hair. And then from a philosophical point, people use these exposome wristbands now. And the idea behind the wristband is that whatever is in the air lands on the wristband, it absorbs, and it represents your exposure. Never really got in the body. It's interesting to me that that's considered exposure, but something that lands on your head and is on your hair is considered contamination. And so I actually think that from a philosophical perspective, they're very similar processes, and we tend to undersell what hair is actually telling us, and perhaps cleaning hair isn't necessarily a good idea. Maybe it actually has some exposure information in the uncleaned hair. If you have plasma, we could also do targeted assays, PFOS, PFOA, PBDE, all of which might be interested for infant cognition. If it's a recently enrolled cohort, you're not likely to find DDT, DDE because it hasn't been used for a really long time, so we would probably steer you away from organochlorines. In terms of targeted assays in your urine, you can measure metals, you can measure phthalates. Certain metals like cadmium are much more concentrated in urine than they are in blood, so it's actually preferred to measure them in urine. And you can also measure cotinine in urine. You can do untargeted assays in the urine, just as you are in plasma, and they are different sets of chemicals. Urine will tend to give you more of the polar chemicals. Plasma will give you the hydrophobic chemicals. So you actually get complementary information actually from doing untargeted assays in both media. Breast milk, you can do untargeted assays in breast milk. You can also measure metals and organics. There's a lot of things you can do, in other words. And what we would do is we would, because we have a budget, we would probably steer you towards what we think would be the most important given the phenotypes and the samples that you have and the sample size. And then there's also teeth. And that's actually what the next example is about. So this is the final use case. So let's say I wanted to do both a prospective cohort study and a case control study. So let's say you have access to a case control study on the genomics of attention deficit hyperactivity disorder. And in your study, there's 2,500 children, 500 cases, and 2,000 controls. And you ran a GWAS on it. You were part of a larger consortium, which everything was pooled, and there were 30,000 people, but the 2,500 are sort of your kids, and you want to see what you can do with environment. And so you come to here and write in an application. You have plasma and urine biobank and stored at -80. And you also have DNA and RNA. So what can we do for you? Well, in our consultation, we would discuss the study design, the demographics, what samples are archived, how long were they stored, were they aliquoted, same questions as we asked a prospective cohort study. But there's some issues because it's a case control design. We can measure samples in urine or plasma, but that's going to be cross-sectional. It's not going to be prospective, unlike the other example. So it won't really be clear if exposure preceded disease or vice versa. Disease can even drive results because kids can be on medications, have unusual diets and behaviors. So we're not really addressing the question of whether or not prospectively early life environments can affect illness later in life. However, there's things that can be done. I'm actually a big fan of case-only designs. So I think the question, does environment modify the disease, is something that we understudy. Maybe kids with autism or ADHD who have higher levels of lead or manganese or methylmercury, maybe they have a more severe disease phenotype because the lead is making their autism or ADHD worse. And that's an actionable finding because you can reduce exposure to lead. So I think those are very, very interesting questions that we don't do enough of. And then you can also look at your control group as a cohort and look at the effects of, say, mixtures on impulsivity. Presumably, you've done some behavioral testing on your controls, and so maybe you can think of them as a cohort. But if you want to look at the ADHD, the original question, the ADHD question, in a case control design, teeth may actually be a way to solve that cross-sectional problem. So, as I mentioned earlier, Manish Arora is a dentist and a chemist. And he's one of our faculty at Mount Sinai. And he developed a method that I think only somebody who is both a dentist and a chemist could have come up with. So it turns out teeth grow like trees. So trees have annual growth rings. And in fact, geologists will actually measure chemicals in those growth rings to tell you whether or not there was a forest fire that year or what was happening in that tree's environment. In teeth, Manish has developed methods using laser ablation and ICP-MS to actually measure chemicals in layers of teeth that he can assign a calendar date to. So he can know the exact week in which these chemicals were actually deposited in the tooth. And those chemicals can include things like lead or manganese, and now he's working on organic chemicals. And he can assign both a dose and a calendar date. And from a statistical perspective, that does two things. It makes your study prospective, even with the case control design, but it also gives you the ability to actually look for critical windows of susceptibility. So this is actually a study that Dr. Arora did with a group in Sweden who had twins who were discordant for autism. So they were identical twins, so genetically they were perfectly matched controls. And one twin had autism and one didn't. And he looked at levels of different chemicals in their teeth, and he can assign calendar dates. So on this graph, the x-axis is age, 0 is the day that the child's born, and the units are weeks, and teeth form in the second trimester, so it goes back about 20 weeks. And he can measure the amount of zinc and how that amount changes over time, but also the association of the concentrations and the disease phenotype and how that changes over time. So this line is basically, think of it as a beta coefficient and how the beta coefficient changes over time. And this is called a distributed lag regression model, which is often used for time series studies, but it can be used with the tooth biomarker fairly effectively. And so what he found is that between the roughly 30th week of pregnancy and about 1 month postpartum, children with autism were more likely to have lower levels of zinc, perhaps slightly zinc deficient, and that is associated with the twin that developed autism. So in this window, zinc correlated with autism, and in this second trimester and early third trimester window, it did not. And after the first month of life, it did not. Although what's happening out here may be interesting in future work. He's also extending the amount of time that he can look at these tooth rings to beyond the first year of life. Right now, it's roughly the first year of life. And then finally, CHEER does a lot of QA, QC. So we're involved in constant, it feels like, proficiency testing and round robin testing. We have methods to reduce batch effects. We put the same sample in all batches, certain numbers of the same samples in all batches so we can adjust for batch effects. We look at pooled samples across lab hubs. So we're trying to adjust for inter-lab variability so that we can actually, if we have a really big job, share across different labs and actually adjust for the inter-lab variability, which is the value of a consortium, because I'm not aware that any other exposure labs do that. We include blinded blanks. The idea behind that is you can be really good at proficiency testing, but if you can't assign the correct value to the correct sample, you've basically just measured noise. So if you put blinded blanks in there, you should get a 0 every time you know there's a blank. And if you don't get a 0, that means something got screwed up in your data processing, going from the measurement to the assignment to the sample. And so we have barcodes and LIMS systems, laboratory information management systems, that help us track that so that that's much less likely to happen. The sort of old school laboratories really use Excel spreadsheets instead of LIMS systems. And I think there were probably mistakes made when people did that. With these new automated LIMS systems and barcodes, it's almost difficult to make a mistake. So you always want to make sure that the laboratory that you're working with uses barcodes and LIMS systems to make sure that the correct result is attached to the correct sample. So in summary, we provide a number of key services, expert consultation. We'll evaluate your bioarchive to help you understand what can and cannot be measured with it. We have state-of-the-art methods for sample processing, tracking, and aliquoting. We do state-of-the-art QA/QC. We have a data center that does statistical analysis on mixtures and metabolomics. Obviously, our goal is to get more and more investigators interested in studying the exosome, which is going to require consortia. It's going to require replication, which is going to be hard because you can't, DNA sequence doesn't change over the lifetime. So you have to factor in when you're measuring the environment as well when you're doing replication in an exosome study, which actually is much more difficult to do and to find a replication cohort. Although it definitely needs to be done, I'm just pointing out that it's going to be a little bit harder. But hopefully one day we'll put exosome on the same plane as the genome. And so that one day we'll be measuring the exosome. And I'm showing you the same picture of Cody because he's the reason why you're here at 1 o'clock listening to me instead of at 2 o'clock. And thank you very much.
Thank you, Bob. That was a great talk. Can you share the slides with us? Because this was really an encyclopedia of information about environmental exposures and how to measure them. That would be wonderful if you could send the slides to Tracy and we could share them around.
Absolutely.
I already have them. Thank you.
Oh, fantastic. Okay. So let me ask people to put their questions in the chat box, and I'll be happy to read them. I'm sure this must have provoked a lot of interesting questions from people in the audience. We had 51 people online, Bob, 51. So, if you'd been here in person, it would have had 100, but.
Thank you for saying that.
Also,
thank you for saying that.
you also can unmute yourself if you'd like.
Yeah, people, yeah, it looks as though if you unmuted, you could ask a question.
Oh, there you go.
One thing of you from Wayne State. Bob, great talk. One quick question. Is there any longitudinal clinical outcome data available for your cohorts, e.g. diabetes, obesity, fatty liver disease, etc.?
I think that question probably refers to the CHEER data center because they are building a larger cohort with information on exposure and phenotype. And yes, yeah, we've done work for the TEDdy Consortium, which is a type 1 diabetes consortium. So we've measured environmental exposures in them. Fatty liver disease, when we switched to here, we got several applications on fatty liver disease. So those are in the works. They haven't been done yet, but that seems to be something we didn't actually do much of, kind of understandably, when we were children's health. And now that we're human health, we've gotten some fatty liver disease studies. So yes, which ones are available? I can't answer. There is an embargo period so that the investigators can publish first on their analyses. I don't think the TEDdy study has yet published, but we did a number of assays for them. I'm actually excited about that one because it's prospective data on type 1 diabetes, and I think that could lead to some really interesting findings, but I don't think it's available yet. They have to publish first, and then the embargo is lifted.
Good. I think we have a couple more questions. Let's see. From Mike Petriello, also from Wayne State. Hi, Bob. Great talk. You mentioned multiple lab measure of PFAS. When we submit an HERE application or submit an ECHO OIF, how is it decided which lab we work with?
It partly depends on where your funding comes from. So ECHO is predominantly Wadsworth for targeted and RTI for untargeted. We are the NCI lab. So even though I don't have a single paper in cancer in my 25 years of doing research, we get assigned all the cancer studies. And there's a NHLBI center. I'm not sure which one it is in all honesty, but it depends in part on which institute funded your study. And it also depends in part on capacity. So NIEHS can be any of the labs because they partly funded all of them. And so we also look at who's the busiest right now, and we often assign them to that.
I'd like to ask a question. I've heard up and down on the NIH program called Children's Environmental Health Centers. They were going to be, not going to be, they are going to be. Do you have any update on that I'm,
Oh, you mean the research translation centers?
Well, it was called Children's Environmental Health Centers, I think. Dana used to have one or still has one at Michigan, and they were under some cloud. They weren't going to be refunded.
They are not. They've not been refunded. There is a new program called Children's Environmental Health Research Translation Centers. So the children's centers were funded for 20 years, and so the feeling was the time was now to translate their findings. So in the new centers, you now have to show how you're going to take research and translate it into action in children.
I see. Oh, okay. So good. So the program remains alive in a sense, essentially.
It's very, very different though.
Okay, different in focus. I have a question from, first I think from Gene Curver from MSU. So interesting facts. At what point can we request a toxicology consult? For example, if we're interested in urinary iodine plus perchlorate, thiocyanate, nitrate, do we need to submit an echo analysis concept first, or can we contact you directly, Bob?
I'm always happy to answer a question. You can email me.
Okay, Gene, you call Bob. I think there's another one. Let me see from Heidi Lemprattle. Now that your research is moving away from children and adolescents and addresses human exposure in general, what would your approach be? Or is there a way to link environmental exposure to aging-related disease like Alzheimer's or Parkinson's? I guess teeth would be the way to go.
Yeah, actually, it's a great question. Manish is working with some neurologists who study ALS, Lou Gehrig's disease. Obviously, adults don't lose their teeth, unlike kids, so you have to pull the tooth. So it's obviously a lot harder. But for diseases in which there are, say, brain banks at death, it's not that hard to pull a tooth. So Manish is getting teeth from people with ALS. I assume that it could be done with Alzheimer's or Parkinson's brain banks as well. So yeah, there are studies of aging. This is not related to cheer and here, but some of our faculty do satellite remote sensing to measure things like air pollution, other geospatial variables. And he can go, Alan Justice is that faculty, he can go back in time in air pollution to when the satellites were launched in the 1990s. So right now, that's not going to work very well for studies in, say, Alzheimer's disease. But eventually, you will be able to reconstruct past exposure as these public databases become more available to a lot of different environmental factors, not just chemicals, over time. So I do think that one day that's going to happen. We may all be retired, or at least I will probably retire when that happens, but I do think that one day that's going to happen.
Bob, dentists have drills, and I wonder if Manish has considered the notion of a dental biopsy so you don't have to remove the tooth.
Well, I have a question for you. Would you volunteer for that?
It depends how good the filling is.
I think of that movie Marathon Man where Laurence Olivier was drilling into Dustin Hoffman's tooth and saying, is it safe, over and over again? So
Very good. I have a question from Vasantha Padmanabhan of the University of Michigan.
Full disclosure, Vasantha once employed my daughter. Thank you. I'm going to tell her that I saw you.
Vasantha is a dear friend of this program, was involved with her cohort, has been involved since the very beginning in our Michigan cohort programs. Can you elaborate on the sexual dimorphism in terms of effect modulation?
Oh, great question. I don't think we do enough work in that area. I think NIH is starting to require it, which I think is great. Here is allowed to measure hormones, so we can measure sex hormones as part of an application to help you get at mechanism of why you might see a sexually dimorphic effect for different chemicals on brain, on behavior, on obesity. To be honest, it's hard to think of a disease in which a sexual dimorphic effect isn't at least plausible.
I'll draw a little bit from a Courtney Carrigan note to Heidi. Some things can be measured using XRF on bone while a person is living. It's non-invasive. Another example of non-invasive measurement. Do you do much of that, Bob?
Yeah, actually, one of our faculty, Andy Todd, has done a lot of that work. He did a lot of work in Korean lead workers in the 1990s. There is a faculty at Purdue that I'm very good friends with.
Oh, yes, that's right.
She's excellent at that. She actually has a handheld XRF that she developed. Yeah. It won't give you timing, which is the big difference between what Manish does, but it will give you cumulative exposure.
Yeah, we talked to the Purdue people, Tracy will remember, when we were working with Flint initially. Nothing actually came of it, but we became acquainted with those techniques. All right, we're approaching 2 and we've taken a lot of your time, and I know you have to get Tony to the doctor. So we'll let you go. Thank you for a very stimulating talk, a lot of good questions, maybe some follow-up. We'll keep in touch. And thanks for everything, Bob. Appreciate it very much.
Thank you. Thank you all so much for having me.
Thank you.
All the best. So now we can move on to part 2 of our program. And part 2 is represents our groupings that we've developed among our Michigan investigators to look at, to consolidate some special interests around nutrition, the environment, inflammation, and one more that's not jumping into my head right at the moment, but I think we begin. I have to bring up the calendar again. Yes, we're going to start with the nutrition interest group, an infant feeding supplement, and everybody has 10 minutes. Maybe you'll get another extra minute because we managed to finish just before 2. So Jean and Gail from MSU, take it away. All yours.
All right, thank you, Nato. Is my sound good?
Yes, very good.
Okay, I'm going to share my screen a moment. All right, we are moving right along without any break here. And I am getting a note, my internet connection is unstable. Can you guys hear me all right?
Yes.
I see Gail nodding. So I am here today to introduce the launching of our Charm Nutrition Interest Group. But before I get to that, I just want to remind everyone that the whole purpose of our Charm interest groups is to foster collaboration amongst ourselves. And where possible, we'd like to focus toward developing ECHO-wide proposals. So just as a reminder, ECHO-wide means any 2 awards in ECHO. So this could be 2 cohorts, it could be 20 cohorts, it could be our cohort along with the DAC or the ProCore, just any 2 awards. And there's no, imperative that that's what comes out of these Charm interest groups. It's just that that's a great way to sort of capitalize on the resource that is ECHO. So that's part of the reason for developing these. We were successful in receiving 3 administrative supplements to our ECHO Parent Award. One is the ECHO COVID NOSI, Notice of Special Interest, that Dr. Sarah Comstock is the PI of that, and she'll talk about that next. So won't say any more about that at this time. However, I just would like to point out that 2 of these 3 awards were ECHO initiated, so the ECHO COVID NOSI, and also the ECHO Diversity Supplement that I'll talk a little bit about in a minute. And then the third one is one Dr. Norman is leading, and she will talk about later today too. And that was not initiated by ECHO, but rather a trans NIH funding opportunity announcement for supplements to parent awards. And this particular one was issued by the Office of the Director, the Office of Research on Women's Health, NICHD, and I believe NHLBI. And it is intended to decrease maternal mortality. And that was a separate NOSI, Notice of Special Interest. So I am pleased to serve as the PI for 2 of those. And the reason I bring them up here is both because I'm about to introduce Dr. Shipp to you, but also because sort of in the spirit of these interest groups, we were able to put these applications together quickly because we had teams of people that had already been working together. So Dr. Norman and I have worked together for many years, previously on the National Children's Study, of course, with Nigel and many of the others of you. And Dr. Shipp, Dr. Gail Shipp and I had worked together on her dissertation. So she and I worked on a project in collaboration with Henry Ford Health System, another one of our Charm partners. And so we were able to quickly sort of segue into writing a diversity supplement for Gail. And so she'll talk a little bit about what her objectives are for that. But I also want to say that she and I will be co-chairing the Nutrition Charm Interest Group. So after you hear a little bit from us, if this is something you're interested in, we would encourage you to contact one or both of us, and we will set up some meetings moving forward. So the Nutrition Charm Interest Group is going to be broadly inclusive of the many, many different components that make up nutrition science. So including, of course, survey information about diet and food intake, but also nutrient levels that can be measured by, as Dr. Wright mentioned, by HERE, either deficiencies or just comparing different levels. For example, zinc, he just mentioned in relation to autism, that would be a great area to pursue. We could look at, back to survey data, things like caloric excess in relation to physical activity or energy expenditure. We have a lot of people interested in obesity work, other aspects of nutritional biomarkers, or social determinants of dietary patterns. So there are lots and lots of avenues, as you all know. And I would just encourage all of you who are interested in any of those aspects to join our group, and we'll see what kinds of things come out of it. In addition to sort of the broad topic areas, there are broad exposure times that we can assess with ECHO. Many of us are interested in pregnancy diet as being sort of a critical window of nutritional exposure that may affect later child health outcomes. Many of us are interested in infant feeding patterns, and others of us are interested in child or family diet. And all of those can be looked at in relation to any of the 5 ECHO health outcomes. So those are that's sort of the broad charts that we have. We don't have anything to present to you about our group yet because we didn't start yet. But again, please contact us if you're interested, and we'll move forward with that. And now I will turn it over to Dr. Ship.
Thank you, Dr. Kerber. So my name is Gail Ship again. I worked with Dr. Kerber in the past, and currently working with her now. I have a background in human nutrition. My doctorate was actually in human nutrition, and I have a master's in nutritional sciences. In terms of contacting me, that is my contact information for moving forward. If interest in the nutritional component of the group. My research interest in relation to ECHO is primarily maternal and child health with a focus on health equity. And then I have a primary focus and interest on breastfeeding in relation to child health outcomes. And that kind of leads me to the next slide, in which the diversity supplement, which was submitted and we received, focuses on 2 objectives. The first one is to look at exclusive breastfeeding and how it protects against risk for childhood obesity among those exposed to maternal obesity in utero. And then the second one, in light of the COVID pandemic, we really wanted to take a look at quantitatively and qualitatively the impact of COVID-19 on women's breastfeeding practices as well as their experiences. So Dr. Kerber talked a little bit about ECHO-wide projects. So my first two aims, currently we're just getting started. For the most part, I have submitted the ECHO concept for the first 2 aims, and those are the titles above. And I am preparing to submit the analysis proposal for the first aim, which is exclusive breastfeeding protective against childhood obesity among women with pre-pregnancy obesity. The last aim, which is focused, is cohort specific. And it does have the potential to inform other ECHO-wide work, but that is a primary, that title and that project is primarily qualitative, in which I plan to explore the decision-making experiences regarding infant feeding practices among ECHO participants who are pregnant or still breastfeeding during the era of COVID-19. And so I particularly have an interest in the COVID-19 hotspot, Detroit, Michigan, as well as other areas to see what their experiences may be, if there were any differences, and go from there. And that's it. If you have any suggestions or questions, please let me know. Thank you.
That's great. Nigel, do you want to, I think we had decided we would keep questions until the end. And so.
I think that's a good idea. And I think we can move right into the next one, which is Sarah Comstock is going to talk about the CHARM microbiota or microbiome working group. Thank you, Sarah. You're on mute, Sarah. You're muted.
Sorry. Thank you so much. My name is Sarah Comstock. I'm an assistant professor at Michigan State University, and I've been working here with CHARM for a while now. And recently I've been in charge of the microbiota working group locally. And so that's what I'm going to talk to you about right now. So just a quick introduction to the priorities for our working group. We are really focused on grant submissions because we are desperately wanting to fund a collection of additional samples that we can use to analyze the microbiomes of mothers and infants in the cohort. And these microbiomes are bacteria, viruses, yeast, fungi that are associated with, typically right now, the vaginal canal, the gastrointestinal microbiome, maybe in the future, the human milk microbiome. We have a lot of interest. And so we've been focused on grant submissions because we would really like to raise some funds in order to do additional sample collections in our cohort. We're also focused on ECHO-wide concepts and analysis proposals because this is the way that ECHO moves forward in the world, is for the ECHO cohorts to work together to produce manuscripts that couldn't be done by a single cohort alone. But it also, we are focused on local manuscripts and projects. And right now we have a very strong team, including myself, Dr. Kim McKee from the University of Michigan, Dr. Lejing Zhang from Michigan State University, Dr. Andrea Cassidy-Bushrow from Henry Ford Health System, Rebecca Nickmeyer from here at Michigan State University, Ting-Fei Ma, also here at Michigan State, Mike Petriello at Wayne State University, and then we've been supported by Tracy Thompson in the study office. Some of our grant successes include the NIH ECHO supplement that Jean Curver just told you about. We are going to be looking at the microbial mediators of the impact of maternal psychosocial stress on perinatal and neurodevelopmental outcomes in ECHO. We are collaborating with 2 cohorts for this, the Dione cohort in Rhode Island and Tom O'Connor's cohort. And we have 2 separate aims. One aim is to understand the impact of maternal SARS-CoV-2 infection, as well as psychosocial stress during the pandemic on the length of gestation. And we are going to look at how the vaginal and fecal microbiomes in pregnancy mediate this relationship using whole shotgun metagenomic sequencing. And then the second aim is focused on infancy, but still understanding the impact of maternal psychosocial stress during this time period on infant neurodevelopment, as well as if alterations to the infant gut microbiome during the first 9 months of life might be mediating this connection between maternal psychosocial stress and its impact on infant neurodevelopment. We've also had some success with applications we've submitted to foundations. So Blue Cross Blue Shield of Michigan Foundation has funded our project, the antibiotic use during pregnancy and antibiotic resistance in offspring. And Dr. Lejing Zhang and I have been working on this project, and we are studying how different antibiotics prescribed and the extent of use of antibiotics during pregnancy might be impacting the maternal and infant antibiotic resistance patterns in their gut microbiomes, and how that may contribute to an antimicrobial gene-resistant reservoir in these pregnant women and their children. Kim McKee, of course, had a vaginal microbiome NIH ECHO Opportunity and Infrastructures Fund project funded for 2019 through 2021. And so she's validating the maternal microbiota signatures in pregnancy with a focus on the vaginal microbiota. And this is a collaboration of many ECHO cohorts. And then we've also had some local CHARM grant successes. So every year the local cohort puts out a call and asks for small projects that might help move along either ECHO-wide or other funding. And so we've been successful in 2017, 2018, and 2020 to get some of those resources for our working group to improve the microbiome resources we have and to make an impact. Currently, we have a lot of pending and in preparation grant submissions. And as you can see, we have lots of members of the team working on these. So it's not just one person. I think one of the things that makes our working group really effective is that everyone is really engaged in what we're doing. And so you can see a lot of different individuals have taken the lead on a variety of other grant submissions, which hopefully will be funding additional work either with samples we've collected already or with giving us the ability to collect more samples from mothers and babies for whom we might already have some samples collected, or to enroll new mother and baby dyads. We're also very busy staying connected with ECHO and ECHO-Wide. And this is how you can find out about these ECHO-Wide concepts and analyses by participating in, say, the Publications Committee or the Obesity Working Group or some of the other ECHO-Wide working groups. This makes sure that we know what's happening nationally, so we know how we can contribute to those efforts. And so right now I am co-leading an ECHO-Wide concept on the early life gut microbiome and childhood obesity. Kim McKee is leading an ECHO-Wide concept related to vaginal microbiota signatures in preterm birth. Kim is also leading her OIF projects, which are considered ECHO-Wide projects, the validation of maternal vaginal microbiota signatures in pregnancy. Dr. Rebecca Nickmeyer and myself are contributing to the writing team for the relationship between gut microbiota and child anxiety and depression in middle childhood and adolescence. And then all of the COVID no-see aims will eventually become ECHO-Wide analyses. So we're very busy contributing not only locally, but also at the national level. And I think that's important, an important way to stay connected with what's happening. We've had some, in prior years, we've had other manuscripts, but just to update you on some things that were submitted and accepted and published this year, we've had 2 papers published this year, one led by myself, Human Milk Feeding Patterns at 6 months of age are a major determinant of fecal bacterial diversity in infants. Another led by Dr. Lejing Zhang, which is Perinatal Risk Factors for Fecal Antibiotic Resistance Gene Patterns in Pregnant Women and Their Infants. We have 1 paper that's submitted currently and 2 papers that are in progress right now. So we're doing a lot, not just at the ECHO national level, but also locally to try to move forward with publications. So some of the keys to our working group success, as I mentioned before, is that the group is really engaged. We're all contributing. It's not being driven by one person. We meet on a regular schedule. So right now we're meeting every other week. If we don't have something as an agenda item, then we'll cancel the meeting for that week. We always work from an agenda, so we know what's going to be talked about at each meeting and can make progress on those projects. We work hard to stay up to date on what the local cohort has and also what our needs are locally in order for us to really contribute scientifically. And this is related not just to the biospecimens that are relevant to the microbiome, but also the metadata that we're going to need to make progress with our microbiome samples. And we're really motivated to secure funds to support additional microbiota sample collections. And this may be the key, one of the keys to why we are so effective, is just because we really feel like the microbiota is pivotal to infant and child development, and that we're not currently funded to collect enough samples. And so right now we're working very hard to get more funds so that we can collect additional samples in order to do that important work. And we, of course, would welcome anyone from this group who'd like to join us. And if you'd like to get more information about something we've done, or if you'd like to get more information specifically about joining the working group, you can email me at comsto37@msu.edu. Thanks.
Thank you very much, Sarah, for this overview, and we'll hold questions for now. We'll now move to what's really our largest and first formed group, which is the Environmental Epidemiology Group, and Mike Petriello will lead our discussion here. Mike, all yours.
Hey everybody, can you hear me?
Yeah.
Thanks, Nigel. So yeah, my name is Mike Petrillo. I'm an assistant professor at Wayne State University. Been there about a year now. And I just want to thank Doug Rudin, especially for getting me involved with this group. It's been really fun. You saw my name with Sarah. I've been part of that working group, and it's just been really amazing for a young person to get to work with all these people. But I'm going to talk about the Environmental Exposures Working Group. I'm a toxicologist, so this is really what gets me super excited. So we're going to talk about our focus areas. Then we'll talk about our grant submissions that we have ongoing. And then we do have some ECHO-Wide concepts analysis proposals that have been accepted, or I guess, yeah, accepted, submitted and accepted, and some that are in the pipeline. We have an amazing group in our working group, and one of the kind of unique, I guess, setups with ours is it's a mix between really well-established investigators like Doug Rudin. You can see the names here, but also some really good rising stars in environmental health. Jackie Goodrich, Courtney Kerrigan, all these people come together, and it's actually really special. So you hear the word environmental exposures. You just saw Bob Wright's kind of presentation. It's such a broad topic. And so when we started this, we were thinking, okay, how can we really organize it and not just have kind of a mess at every meeting? And so early on, we decided to figure out 4 or 5 kind of focus areas based off of who is actually on part of the team. So it's come to focus area 1, which is PFAS, exposures and associations with birth outcomes. Focus area number 2, which are VOC exposures, so volatile organics, and specifically analytical challenges. We have 2 really good analytical chemists on our team, Stu Batterman and Paul Stemmer. Focus number 3, pollutants of current CHARM funding. So if you go back and read the current grant that we have, there's a lot of talk about phthalates and metals, and that's being led by Doug Rudin. Focus area number 4 is kind of really interesting, especially with COVID right now. Interactions between chemical and non-chemical stressors. So we gave the idea of, okay, what if you're exposed to COVID, but also some sort of chemical pollutant? Is that something we can look at? Or what about diet? So we heard about the diet working group. That's one thing about these working groups. I think it's going to be a back and forth. A lot of us are going to be on multiple working groups. So some of the other grant submissions that we're working on now, I have a local OIF resubmission. I guess I should have put RE in front of that. We also have a Victor going in. So this is a virtual consortium. It's a NIEHS grant. And the whole idea is to bring groups together that haven't published before. And so we're focusing on different labs and different researchers in the Midwest. And a lot of us are part of these working groups. So just to go a little bit more specific about these grants. So this is the OIF that I'll be resubmitting. It's pretty much exactly the same, except for one caveat that I'll kind of point out. So the title is Impacts of Interactions Between Environmental Chemical Exposures and Diet on Gut Microbiota and Microbiota-Derived Metabolites in Moms and Kids. And so this time around for the OIF funding, they really are interested in interactions. And it just lucks out. That's kind of what I did throughout my whole PhD. I looked at what happens if you eat a dietary stressor, but are also come into contact with a chemical pollutant. So it's kind of perfect for this OIF. But so the aims, they're written there, you can read them. But the overall idea is to kind of look at this idea that maternal exposures to a chemical pollutant, we're focusing on PFAS because it's of all the chemical pollutants that Bob Wright kind of looked at, they're probably the highest body burden in all of us right now. That are also linked to adverse health outcomes. We're interested in modulation of maternal gut microbiota and the metabolites that those microbes create. And our health outcome is primarily going to be decreased infant birth weight. And then aim 2, we're really lucky. Kim McKee has an interesting sub-cohort where they're actually collecting fecal samples from kids over time. So we're going to kind of leverage those samples, but also Andrea Cassidy Bushrow at Henry Ford has another study that has even more samples. So this is what we kind of sent out for the first OIF. The new kind of cool thing that we're doing, we're going to collaborate with Ann Dunlap in Atlanta, and she has hundreds of samples that are similar in scope of this with from Atlanta individuals. So the very different kind of population that would be at Kim McKee's Ann Arbor folks. So this is the outline of the Victor grant that we have going on. So remember, it's a virtual consortium. So that's actually a very important part of it. So we're all experts in Zoom now. So I guess we're all really good at virtual consortiums. So we're bringing together three primary PIs, Andrea Cassidy Bushrow at Henry Ford, myself at Wayne, and then Mark Brown at the Cleveland Clinic. And so we have an epidemiologist, we have a toxicologist, and we have an expert in the gut microbiota. And so with this team, we can study associations in human samples, but also kind of test mechanisms using mouse models. So that's what Mark and I do really well. But you can see, so those are the primary PIs, but if you look to the right there, this is a huge group. And a lot of these people are on our working group. They're at University of Michigan, Michigan State, and Wayne State, Henry Ford. So we're really excited for this submission. And the main outcomes of this will be cardiometabolic outcomes. So the idea is if a mom is exposed to a PFAS or some chemical pollutant, does that kind of predispose the offspring for cardiometabolic disease? So we're really excited about this grant. Okay, so now we'll talk about some of the analysis concepts that have been accepted and I guess submitted. This one was from Jackie Goodrich. She's University of Michigan. So we have to have a shout out to Dana Dolinoy for kind of suggesting Jackie. Thank you, Dana. So this is kind of an outline of her proposal. You can read the background. You heard a lot about PFAS already. But so the concept is actually to address the hypothesis that gestational exposures to PFAS will be associated with altered DNA methylation in newborn samples and genes relevant to early life growth and metabolic programming. And so what's cool is we're going to be working with cohorts such as ARCH, which is ours, the MMIP, which is part of University of Michigan, Marbles, and then that same Atlanta Echo cohort. So we're excited to get this started. The other one was set up by Doug Rudin, and it was kind of an interesting path to get this accepted. This was through that speed dating mechanism, if you read about that in the Echo newsletters. And so Doug's really interested in phthalate exposures and autism spectrum disorder. So what's cool, you can read the objectives and everything there. But what's cool about this group, this application is we're working with at least 2, if not 3, additional cohorts across the country. And so this one was accepted, and so we should be working on that one soon. And then I have one that I've been working on. I've been thinking how best to kind of submit this because it's kind of related to my OIF. So I'm probably going to end up doing it in parallel. But so what happens when we were looking for other cohorts to kind of collaborate with, very few actually had chemical exposures and 16S or stool samples, but a lot of them actually have metabolomics or are going to have metabolomics in the future. So echo-wide, we can kind of test the hypothesis that PFAS exposures in the moms can actually modulate the gut microbes' metabolites, kind of give us more evidence that PFAS may be playing a role in microbiota disruption. And so far we have CHARM groups working on this, that Atlanta cohort, and then one from Colorado and one from Kaiser Permanente. So we're excited to kind of submit this as well. But like Sarah said, we're open to new members. If you're interested in environmental health and chemical exposures, you can email me there. With that, thank you.
Well, thank you, Michael. That's a lot going on in the environment. I just would suggest as a perinatal epidemiologist that not to use birth weight just as a outcome, but consider gestational age and fetal growth because the two are intermixed. But I'm sure that's easy enough to do. So just a thought. So we are going to move on to the next group, which according to my list is the inflammation interest group. And we have Karen Rasico and Miata from Michigan State and Miata Buxton from the University of Michigan. And I believe I saw Reina Ficharova in the background too of this meeting. So I'm sure Reina may be very interested in this. So I guess we'll start with Karen. And Karen, are you here?
I am here.
Good, take it away.
Alrighty. And let's see. Are we all seeing my screen?
Yes.
Yay. Okay, I promise that's the only awkward question I'll ask everyone with the mutes on. Yeah, so I'm Karen Rasko. I'm an assistant professor at Michigan State in OBGYN. And so Jean kind of started us off with the nutrition group, which hasn't really begun yet. And I am going to wrap it up with the inflammation group, which has kind of begun, but we're still a little bit of work in progress. So basically a few months ago, we kind of initiated this group. This is kind of our little blurb that we sent out to everyone, similar to what you've heard from my colleagues. This, is hopefully going to be a platform where investigators from different disciplines and career stages can meet and collaborate to work on, really, again, the goal is research, funding opportunities, and kind of a way of getting together to put together some of these ECHO-wide projects. And so I started putting together, or trying to kind of think about how we were going to put together a working group for inflammation. And I think that probably one of the things that we have been dealing with is similar to what Mike had mentioned before, is that this is a really broad research area. And so basically we've been trying to kind of think about a way to organize this group where it is a supplementary group to those that are already out there. And so what I mean really is, so I have on this slide kind of an example of one of these charts. And we had one meeting so far with the inflammation group. And what we found, what I noticed was that, so first of all, we have a number of really awesome people who are on several of these working groups. And everyone who was at this meeting was also a part of at least one other working group, right? And this really is because in our field and in our group, inflammation, kind of what I have found, seems to be something that almost all of us are studying, and most of us are studying as an outcome that is downstream of the main exposure that we're experts in, right? So the environmental chemical groups, they're all meeting and studying the effect of these chemicals on outcomes, many of ours, which are, birth outcomes and behavior outcomes. And downstream of the chemical exposures, many are suggesting that inflammation is a mediator of our outcomes. And I found that this also seems to be the case in so many of our colleagues in the microbiome group. And I'm guessing in the nutrition group, that might also be something. So this is kind of something that we noticed in the first group, and we've kind of gone back and we're trying to, brainstorm ways of making this working group, either effective on its own and having kind of its own presence that isn't kind of a mediator type topic for all the other groups, or maybe we'll just do that. So what I kind of chose to do today, so like I said, we talked about this, we met once, and that's where we kind of found, well, this is kind of, it might be a little bit redundant, it might not. Maybe we just need to organize it differently. But I've kind of wanted to put a couple of my ideas out there, and everyone, I know you're banned from asking questions till the end, but these are just two ideas that, if you guys are hanging out sometime and really have nothing else that you feel like doing at the moment, if you don't mind pondering these ideas for us, we would love anyone's input. So a couple of things I was thinking about how to make this either not redundant or kind of work for a group is, first of all, something we kind of discussed when we were actually at our first meeting was making this maybe a working group that really is independently focused on what I study, and I might be probably close to one of the only ones, looking at infection-associated inflammation. And when I say that, I mean acute outside of the microbiome. And again, ECHO, when I talk to the officers at ECHO, it's not wanting to focus on infections, but then COVID happened and they said, oh no, we're focusing on infections. So this might be something that more people are interested in, that, we might actually be able to gain some resources from ECHO-wide mechanisms. And then of course, my other proposal is kind of what I've been, doing quite a bit in the last year or so myself, is working more as like a panel or a group of individuals who have this expertise, like not primarily in some of the other exposures most of y'all are working on, but maybe a group of reproductive immunologists such as myself who study immunology primarily that could, kind of like Sarah mentioned, come to meetings for the other groups or our own on a as needed basis for when people need a consultation or someone who can work with them on their proposals as an expert in inflammation. And I've done this with many of you, my colleagues on their grants. Happy to do it. Really exciting science that I'm reading, and definitely can see where inflammation fits in. So basically to summarize, I'm thinking and would love anyone's input of, yes, trying to kind of identify a group that wants to work more on inflammation, acute type infections and inflammation associated with that, and or working kind of on an ad hoc basis, having kind of a group of immunologists, people, again, Raina, myself, others who are, study infection or inflammation primarily, that might be able to meet with the other working groups on an ad hoc basis, or even, that, again, other groups could kind of invite based on what their agenda is. And so that is really where we're at with the inflammation working group. And I was just going to share just one last slide. We also have one, just as an example, kind of what I say of acute infectious related inflammation. The one ECHO wide proposal that we have out right now is focusing on a infectious exposure, which is maternal CMV, and looking at how potential inflammatory cytokines are mediators of the relationship between maternal CMV during pregnancy and SRS scales that are measuring ASD behaviors in children. And so this is one example of what I mean by, if we have others that are interested in infection-related exposures related to outcomes, this is kind of an example of one of the proposals that we've put in already. So with that, I will wrap it up, and we'll move on to our placenta inflammation discussion.
Nigel, you want to introduce Miata? Dr. Buxton?
Well, Miata, you can probably introduce yourself, Miata, but I will say that you are a scientist at the University of Michigan who we've been working with for some time. And we've been very impressed with what Miata has been doing. And if you could say a little bit about yourself, Miata, that would be helpful. You're good to go. We
have to unmute.
You have
Yeah.
to unmute, I think.
Thanks, Karen.
Still not hearing you, Miata.
Miata, you're still muted. Mark, can you see? Oh,
Can you hear me now?
you got
Yeah,
it.
that's working.
Thank you.
Now we'd have to go back to your slide. There you go.
Okay. Thank you. Thank you. And thanks, Nigel. So I am Miata Buxton. I am an assistant research scientist at the University of Michigan in the epidemiology department. I am interested in maternal and child health outcomes, in particular, some of the environmental and dietary exposures that could impact maternal and child health outcomes. And I'm also interested in the mechanistic pathways. So that includes inflammation between a number of these exposures and outcomes. So I'm happy today to share with you our pilot project, which we hope to get started soon. And hopefully we can get some feedback from you and potential interest in terms of collaboration going forward. So I will be sharing early plans on our pilot, which aims to investigate the association between particulate matter and placental inflammation, and also trying to focus on understanding the role that maternal nutrition could play in this association. So I want to start first by acknowledging collaborators and, of course, CHARM which we will be using data from March, so placenta samples and questionnaire data. And also we have received funding from the University of Michigan MCubed program. And I would like to take this moment to recognize my collaborators, who are from a number of institutions. So we have collaborators from MSU, other collaborators from the University of Michigan. We have Reina from Harvard, and then also Caroline Talafia from Placental Analytics. So really multi-institution project, and we have now had an opportunity to grow this project from here to an ECHO-wide concept, which also includes people from other institutions. So definitely excited about this project and other potential opportunities to expand this. So the motivation for this project is that we know that environmental exposures such as airborne particulate matter, so this really refers to a mixture of solid particles and liquid droplets that are part of air pollution. These are hypothesized to be risk factors for placental inflammation. And this happens because you have, after inhalation of pollutants, so airborne particles, you have inflammation that could occur in the lungs. And based on that, the inflammation there can become systemic and then can then travel to the placenta. The other way that this could potentially happen is that for some of these particles that are really fine, you could have those particles actually traveling from the lungs, and then those particles are able to cross the placenta barrier, and then thereby leading to systemic inflammation at the placenta level. So with this, we are interested in exploring this association using CHARM data, but then we are also interested in looking at maternal diet, because we know maternal diet is something that's modifiable. And we know based on recent information using the dietary inflammatory index that it could contribute to systemic inflammation. And we are interested in understanding whether this could potentially modify the association between air pollution and placenta inflammation. So part of this study, and based on this, we are interested in using the dietary inflammatory index. This is an index that was developed by Shivappa and James Hibbert from the University of South Carolina. And this index quantifies the inflammatory potential of the diet. And we are going to be able to use this based on the dietary information that CHARM is collecting as part of the study. So in particular, we're hoping to evaluate the association between the dietary inflammatory index and placental inflammation. We are also looking to evaluate the association between the estimated exposure to daily outdoor PM during gestation, and then also accounting for spatial variation, which I'll speak about a little bit later, and then and placental inflammation. And then finally, we will be looking to evaluate if air pollution associated placental transcriptomes signatures are modified by the consumption of diets that may be pro or anti-inflammatory. So some of the innovations associated with this project is that we're using the dietary inflammatory index. Again, this is this index that offers an opportunity to not just think about diet in terms of individual foods consumed or dietary patterns, but specifically the potential of the foods eating to contribute to systemic inflammation. So this is really a very important step that we are hoping to capitalize on this pilot. And of course, hoping with some interest, we can get some other investigators from other studies who may have the types of data that we need, and then see if we can combine such information as a next step potentially. And then the next step here, the next important innovation in this study is that we're using the Global Transcriptome ESH system. And this system is really used to be able to sequence mRNA associated with placental inflammation. The plus we're using this system is that it requires a low amount of samples to be used, and which is really critical and important for these types of studies that are designed to conduct multiple secondary data analysis. In addition to this, we also have the advantage of having the system being able to be sensitive even for samples that have varying degrees of quality over time. And we know this is a long-term study. So this system is going to hold up well based on the ability to be able to still accurately produce results based on samples that have varying degrees of quality. And then the final part about this study is that for air pollution exposure, we have studies that have looked at air pollution using citywide average. And citywide average precludes the ability for you to be able to actually understand how close a person is from the source of the air pollution estimate and how accurate or how much of an improvement that gives you if you're not using the individual's address. So for this study, we are going to be able to incorporate participants' address into estimating the exposure for each person. And this adds to improving the air pollution estimates for each individual because we are incorporating the distance a person lives from the monitor that produced the estimate. So again, we are open to questions and suggestions and any potential opportunities for people who have the types of data that we may be using and to see how we can combine these different types of data sets going forward, especially past the pilot phase of this project. I also want to note that, I think I said it a little bit at the start, that we have individuals from this group who are working with the ECHO-Y analysis looking at dietary influences on maternal inflammation and perinatal outcomes. So just to share that as well. I have my contact information here. So if anybody has questions or anybody who's interested can feel free to contact me. So with that, I will end my presentation.
Thank you very much, Miata. That was very interesting indeed. We're holding questions for a little while longer. We have one, our fifth presenter today, or fifth topic, Gwen Norman, a dear friend for many years, worked on the National Children's Study with us from Wayne State and has received a special supplement that Jean mentioned briefly, and I'm going to let Gwendolyn tell you all about it. Gwendolyn, you're on. I'm not hearing you if you're talking. I don't know if you are.
Hi, I am talking. I'm here. Thank you. I want to thank you. And it's really my pleasure to present this study, even though the reason for this research is really not pleasant at all. But I cannot discuss this subject without telling how extremely grateful I am to Jean Curver for her invaluable assistance in getting us to this point. When Nigel sent the notification for the supplement to me, we only had about a month to get everything ready and completed and submitted through 2 universities. And Dr. Curver worked with me day and night every step of the way to make sure we successfully made the argument for this approach and facilitated the collaboration between both Michigan and the Georgia cohorts. So thank you, Jean, for getting us to this point. But I'm going to move forward. As I said, this is not a pleasant subject. Let's see. This is not. Okay. So in April of 2016, Kira Dixon died after bleeding internally for more than 10 hours following routine C-section. She left behind a husband and 2 sons. And Charles Johnson, who is the husband of Kira, founded For Cura for Moms in Atlanta. You probably heard about this. Those of you who are associated with NIH, Dr. Shalon Irving, 36, died January 28th, 2017, 3 weeks after giving birth to her daughter, Solel Irving. She was an epidemiologist at the CDC and researched how structural inequality affected health outcomes. And Wanda Irving and Bianca Pryor, the mother and best friend of Dr. Shalon Irving, are the founders of Dr. Shalon's Maternal Action Project, another one coming out of the state of Georgia. Then Amber Rose Isaac this year on April 21st, and I followed this in the news. It was in the height of all the coverage of the COVID pandemic and the deaths. She tweeted on April 17th about her negative experiences at Montefiore Medical Center in the Bronx. And Bruce McIntyre, her partner, founded Save a Rose Foundation in the Bronx in New York. Yolanda Shifra Kadima, 35, died July 27th of this year, 3 days after a C-section delivery of her twins. She leaves behind 7 children, including her newborn twins. But the tragedies continue, and I don't know how many of you have saw this. Last week on November 2nd, there was a news announcement that said members of the medical community in Indiana mourn the loss of Shanice Wallace, MD, a 4th-year pediatric chief resident there after she died from pregnancy complications last week. At about 35 weeks into her pregnancy, she developed preeclampsia. She was delivered by C-section, but continued to have complications, which included a ruptured liver, kidney function issues, high blood pressure, and required additional surgery. She died on October 24th after her daughter, Charlotte, was born. And this was the headline, Black Doctor Dies After Giving Birth. One of the things that connects most of these stories of these women who were sophisticated, well-educated, were able to speak for themselves, is that they complained that something was wrong, that they had an issue that was not addressed. So the supplement, just really quickly, the supplement background, 700 women die each year from complications related to pregnancy. About 50,000 experience severe maternal morbidity. And so the NIH has a new initiative, which is called Improve, which is implementing a maternal health and pregnancy outcome vision for everyone. And the idea of Improve is to support research on how to mitigate preventable maternal morbidity and maternal mortality, and decrease severe maternal morbidity and promote equity in the United States. And there were several goals. I'm just going to highlight the first, which is the one we selected, which was to incorporate community partnerships and participation in domestic maternal morbidity and pregnancy-associated morbidity, domestic maternal mortality and pregnancy-related morbidity research to resolve health disparities and attain equity in maternal health. There were 15 areas of interest. We selected to develop strategies for enhancing community partnerships that engage women and their families, communities, healthcare providers, and systems and local governments to identify barriers to and community-specific opportunities for optimal maternal health equity. The rationale is we're here still in the midst of COVID, and we know what we can do to protect ourselves. We know that we can do hand washing and wear masks and socially distance that can protect us. But in contrast, there's very little known about how to ward off the impact of bias and racism and how it gets under our skin. And while the most common causes of maternal health, such as infection, heart conditions, hypertension, bleeding, strokes, can be prevented or at least mitigated, this opportunity is compromised when mothers do not receive equitable care and when moms avoid healthcare services rather than undergo the stress associated with adverse and unfair experiences. So our overarching goal is to empower women by discovering strategies that we can employ to lessen the impact of unconscious or implicit bias in the healthcare setting and thus reduce the risk of severe morbidity and mortality. So just to emphasize why this implicit bias has such an impact is the attitudes and behaviors of healthcare providers have been identified as factors that contribute to health disparities. In particular, in 2003, the IOM said that unrecognized bias against racial and ethnic minorities may affect communication with providers in the receipt of care. And most importantly, this showed that implicit bias was significantly related to patient-provider interactions. In fact, implicit bias was more often significantly related to patient-provider interactions and health outcomes than to the actual treatment processes. So in summary, a critical examination of U.S. maternal health disparities support roots in Black women's exposure to systemic racism, with Black women being more likely to be uninsured and receive less preventive care, to die of pregnancy-related conditions at a higher rate than white women with the same condition, to receive lower levels of obstetrical care based on their location, and to experience racism and implicit bias within the healthcare system. So the goal of the study is to conduct community-based participatory research to identify interventions to reduce the impact of bias in healthcare that contribute to increased risk for maternal mortality and morbidity in high-risk populations. And this study will therefore engage African-American mothers, community maternal health advocacy organizations, and healthcare providers in a series of structured interviews and focus groups. Utilizing a mixed method approach and doing content analysis, the data obtained will be used to develop a toolkit of specific strategies for mothers to counter negative interactions and to obtain patient-centered, respectful care. So the first specific aim is to identify facilitators and barriers for mitigating the impact of implicit bias and racism on African-American women in the healthcare setting based on input from key informants. So we're going to conduct virtual focus groups with women who are participating in the ECHO to understand their experiences in seeking prenatal delivery and postpartum care. We're also the Atlanta ECHO group is also going to be conducting parallel focus groups, and both sites will conduct structured interviews with healthcare providers from the health systems where participating women have delivered to gauge their knowledge of the role of implicit bias and racism on African-American women's prenatal delivery and postpartum care. The next aim is to develop strategies that will optimize utilization and effectiveness of healthcare services for African-American mothers. So there will be a second round of virtual focus groups and interviews with the same African-American mothers and healthcare providers identified in AIM 1, and will solicit input for developing an intervention at supporting changes needed to end the implicit bias and systemic racism that African-American mothers face in accessing delivery and postpartum care. And we will evaluate healthcare providers' experiences with training simulations around implicit bias, and all activities will be completed in parallel with the Atlanta site. Okay, so I'm just going to go to the study questions, which are: what are the facilitators and barriers to the equitable and respectful prenatal delivery and postpartum care for African-American women from the perspective of African-American women? And to what extent do Black women's healthcare providers acknowledge the role of racism and implicit bias in the healthcare of African-American women? And the phase 2 questions will be: what do African-American women see as strategies for improving their utilization of and engagement in prenatal delivery and postpartum healthcare, and their ability to adhere to treatment recommendations? And then, do providers' perceptions of the roles of systemic racism and implicit bias in medical decision-making change following a training simulation? So we have our partner stakeholders are the IHI Merck and the Southeast Michigan, SimQuick and Southeast Michigan Perinatal Quality Improvement Coalition, because we recognize their alignment with shared missions surrounding equity and birth outcomes and their unique contributions to each respective organization. I think I'm running over my time a little bit, so I just want to say thank you. If you have any questions, we can move ahead with that.
Thank you very much, Gwen. You've heard a lot of wonderful presentations. We have just a little bit of time left. I would say that, unless you have a very pressing question, you have everybody's email addresses, and we have all the presentations. Tracy has them, and we can send them to you. I will just read that Raina just said how much she has been energized from all she heard today, and she has to leave, but she will certainly be involved in our immunology inflammation work. So this has been really a wonderful experience. I hope you can send your questions in to the presenters because we lack time. I just do want to quickly mention that we will have a few seminars coming up in 2021. In particular, on January the 14th, Greer Page, statistician from Research Triangle Institute, will talk to us at 2 p.m. You're getting getting notices about that. And most recently, I've been able to talk to 4 different outstanding ECHO people to talk about each of the 4 ECHO outcomes: obesity, pulmonary issues and asthma, perinatal issues, and neurodevelopment. And we'll be sending you notices about those seminars. So we'll keep the intellectual life of CHARM alive. I should have shown a slide about CHARM. CHARM is our group, Child Health Advances from Research in Mothers. That's who we are. We're a universe of scientists and who are interested in how we can study child health and maternal health through incorporating studies of both mothers and children together. And we're focusing on both mothers and children. And we have cohorts and we have grants, but CHARM is the all of us.